So far, we have talked about the first two steps on how pathogens are able to colonize a host, which can lead to infection and disease. So, step one was to maintain a reservoir, and step two was to gain access into the host, which for human pathogens, that would be getting into the human body via their portal of entry. And we also had to consider the infectious dose. So for step three, now that the pathogen has entered the body, it needs to adhere or attach to the particular body surface so that it can then invade the specific cell or tissue type, okay, that it needs to infect. So bacteria and viruses have different strategies for adhering to different cells. So when it comes to bacteria, one approach that they use are little molecules called adhesins. So they're called adhesins because they help the bacteria adhere or attach to the cell. And you can find these little adhesin molecules on the capsule, which is that sugary layer that is outside of the cell wall if the bacteria have a capsule. Pili, which we talked about with conjugation, also contains adhesins, as does fimbriae. So remember, fimbriae are those little short hair-like projections that help bacteria attach to things, like body surfaces. And so you can see in this picture that E. coli cell has a few pili, and on the end of them are those little red dots, and those are adhesins. And so they're going to allow the E. coli cell to adhere to those different receptors on the surface of the host cells. Another approach that bacteria use for attachment are biofilms. And so remember, this is caused by the presence of that capsule, again, that sticky, sugary layer. And so biofilms are essentially community or a community of organisms, okay? Different types of organisms, as you can see there. There's little yellow ones, and then there's blue ones with flagella. So you'll have your kind of colonizer organism that kind of originally absorbs to the surface, and then other organisms kind of join them, and they eventually secrete this sugary matrix that kind of encapsulates them, and it actually protects them as well from things like antibiotics and whatnot, because it's hard for the antibiotics to penetrate that sugary secretion. Okay, so these biofilms are sticky and help the bacterial cells adhere to different surfaces. Viruses really have one main approach, and those are those spike proteins that we talked about a few weeks ago. We're also referred to them as docking proteins. And so that is, as you can see in the picture, the docking proteins or the spike proteins are either found on the capsid of the virus, or if it's an enveloped virus, you'll find the docking proteins there. And it's just going to attach to a receptor molecule on the host cell. Okay, and then from there, once they attach, they're essentially going to trick the cells into letting them in. And so that's going to be step 4, which is going to be invasion of the host. And so there are a variety of ways that microbes do this. One is through the presence of enzymes, sorry. And so there are a variety of enzymes that we're going to talk about in the future that allow bacteria, for instance, or other microbes to essentially escape host defense mechanisms so that the pathogen can gain access to an environment that's very rich in nutrients and where they won't have to really compete with the normal microbiota for resources and space. And so one example of such an enzyme is hyaluronidase. And hyaluronidase is an enzyme that is going to digest and break down hyaluronic acid, which you may have heard before if you've taken anatomy and physiology. Hyaluronic acid basically acts like the glue that is going to hold cells that make up tissue together. Okay, and so you can see in this picture, the left and middle cell they have that purpley glue, that's the hyaluronic acid, that's forming a nice tight seal so that bacteria and other pathogens can't penetrate through. But some bacteria have an enzyme that will break apart that glue, giving them access to the area below. So that's one approach for invasion. A second approach is going to be more specific to intracellular pathogens. So intracellular means that they live and replicate inside of a specific cell. And so a virus is a good example, right? They're intracellular pathogens. And so they have to get inside of a cell where they then hijack host machinery and take over. There are some intracellular bacterial pathogens as well. And the other picture in your PowerPoint is chlamydia, which has a really unique life cycle that we'll talk more about later. But it is an intracellular pathogen as well. And then some pathogens are really sneaky, and they use what we call the Trojan horse approach to getting into those deeper tissues. And so, for instance, some pathogens are going to intentionally be phagocytosed because they are going to be able to avoid digestion. Because as you'll learn about in our next section of this course, innate immunity, macrophages are a type of phagocytic cell, and they will chase down the pathogen, and then they will ingest it, as you can see in the picture on the far right. And so the pathogen that gets ingested is placed in a phagosome. And eventually that phagosome, which is harboring the pathogen, is going to fuse with my favorite organelle, the lysosome, and the lysosome is going to essentially destroy it, right? Because it contains those hydrolytic enzymes that will break down the pathogen. But some pathogens are sneaky. Once they get ingested and are in the phagosome, they are able to break free from that phagosome before the lysosome even gets involved. And so they're able to replicate in there, and they actually just use the macrophage kind of as a transportation device so they can get to other parts of the body undetected. Okay, so that's why we call it the Trojan horse. All right, that is going to be the end of this video. Please let me know if you have any questions.